Most of what we find inside of our skeletal muscles are the cells called skeletal muscle fibers. Skeletal muscles, as we can see in the picture here, have that tube within a tube plan, right? So the big tube is the skeletal muscle, but that's broken up into smaller pieces called muscle fascicles. And then inside the muscle fascicles, we have the smallest tube, and those are the muscle fibers themselves. When we look at a cross-section of the muscle fiber, we likewise see a tube within a tube within a tube plan. The smallest tubes in the skeletal muscle fiber are called myofilaments, and these are just the proteins that contract to cause the muscle to move. Myofilaments are bundled into groups called myofibrils. You can think of myofilaments as being like sticks, and then you want to bundle your sticks together because it's easier to carry them. The bundle of sticks would be the myofibril. And so we have lots and lots and lots of these medium-sized tubes, these myofibrils, inside of the larger tube here, which would be the skeletal muscle fiber. The myofilaments, or proteins in the skeletal muscle fiber, are organized into structures called sarcomeres, which I talk more about in another video. But I wanted to mention them here because the sarcomeres contain a very high organization of proteins. So the proteins are organized in such a way that sometimes they're overlapping and sometimes they're not. In places where they overlap, it looks dark, and places where they don't overlap, it appears light. So when we look at these skeletal muscle fibers with a microscope, we can see this light, dark pattern, and this is the organization of the proteins in the muscle fiber, specifically in the sarcomeres of the muscle fiber. Cardiac muscle tissue also exhibits these dark light patterns. We call these striations, by the way. But smooth muscle does not have striations because it doesn't have a high organization of protein like cardiac and skeletal muscle. Now we're going to talk about the important organelles associated with the skeletal muscle fibers. Sarcolemma is just a fancy name for the cell membrane of the muscle fiber. Sarco and myo are both prefixes that mean muscle. Lemma means cell membrane. So literally this just translates as the cell membrane of the muscle fiber. Transverse tubules, or T-tubules, are parts of the sarcolemma that transverse the muscle fiber. And what this means is that in some places, the sarcolemma dips and goes in deep to the muscle fiber, then comes back up, and then it dips in and goes deep to the muscle fiber, then comes back up. So places where the sarcolemma invaginates, we call those the T-tubules. And both the sarcolemma and the T-tubules will be very important for skeletal muscle physiology. We know cytoplasm is everything we find inside of the cell except for the nucleus. So sarcoplasm is simply the cytoplasm of the skeletal muscle fiber. So all the stuff in there, the myofibrils, the myofilaments, any organelles, it's all that stuff except for the nuclei. In our cells unit, we talked about an organelle called the endoplasmic reticulum. And the endoplasmic reticulum could have different functions like detoxification or making proteins or storing calcium. The sarcoplasmic reticulum is just a type of endoplasmic reticulum that stores calcium for the muscle fiber. You absolutely have to have calcium for muscle contraction to occur. It's critical, and we talk about this more in another video. Skeletal muscle fibers also have many mitochondria inside of them. And that's because the mitochondria are the organelles that are the powerhouse of the cell. They're making the ATP, the energy that's needed for muscle contraction to occur. Muscle fibers are also multinucleated, meaning you find many nuclei within one single muscle fiber. And this is because of the way the muscle fiber develops. We start off with one little muscle cell with its nucleus, and that merges with a second muscle cell and its nucleus. So you get one larger cell from the merging, and it retains the two original nuclei. And then that cell will merge with a third cell, and we keep retaining the nuclei. We keep doing this process over and over and over again, so we end up with one really long, thin cell retaining all of the original nuclei of the original cells.